Welcome to the PubMed Daily Review for March 30 SD. Today we scanned 1697 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, targeted cancer therapies and reirradiation. This trend focuses on precise cancer treatments, including targeted drugs and reirradiation, to improve patient outcomes. This cluster covers 214 papers. Representative work includes RGX-019. MMA inhibits leukemia progression by targeting MER proto-oncogene tyrosine kinase, MERTK, in acute myeloid leukemia, correction, reirradiation and oligorecurrence, and oligometastatic cervical cancer in modern radiotherapy era, and retraction note. A water-soluble nucleolin aptamer paclitaxel conjugate for tumor-specific targeting in ovarian cancer. Trend 2 of 5. Food science and metabolism research. This trend explores how biological processes and molecular mechanisms influence food characteristics and metabolic health. This cluster covers 458 papers. Representative work includes advances in wine yeast autolysis, biochemical and molecular mechanisms, and a release of organic compounds in white and sparkling wines. An updated review, steak dry aging. An innovative approach to producing high-yield, high-quality dry-aged beef, and anti-lipogenic effects of iridate-rich Lonicera caerulea berry extracts in a cortisol-stimulated adipocyte model. Trend 3 of 5, liver disease and biomarker discovery. This trend focuses on identifying and validating biomarkers and clinical markers for understanding and diagnosing liver disease and related conditions. This cluster covers 356 papers. Representative work includes clinical outcomes and non-invasive testing in metabolic dysfunction-associated steatohepatitis with cirrhosis. A systematic review, the conjugated bile acids profile, suggests a novel liver muscle axis associated with sarcopenia in chronic liver disease and exploring heart rate variability as a biomarker for frailty. A narrative review. Trend 4 of 5, mental health and social resilience. This trend explores how individuals and communities build strength and cope with challenges, encompassing mental well-being, social support, and adaptive strategies. This cluster covers 380 papers. Representative work includes game changers in the treatment of and engagement with persons living with mental health and substance use disorders. The role of the nurse, rethinking social capital in wildfire resilience. The case of Central Portugal, and what tools, technology, and techniques enable participation in an e-making program for people with intellectual disabilities. Trend 5 of 5, AI, genomics, and motor learning. This trend explores AI's role in analyzing biological data and improving motor skills. This cluster covers 289 papers. Representative work includes investigating the readability and quality of AI systems to trending questions about food poisoning, optimum recombination rates for genetic gains and simulated recurrent selection in empirical maze populations, and toward next-generation machine learning and deep learning for spatial omics. Up next, our first paper out of 5, Combo, a combined human bone and lympho-myeloid bone marrow organoid for preclinical modeling of hematopoietic disorders by Yuqi Shun and Abdullah O. Khan from Molecular Hematology Unit, Weatherall Institute of Molecular Medicine, Radcliffe Department of Medicine and Institute of Health Research, NIHR, Oxford Biomedical Research Centre of Oxford, UK. Let's break down its key contributions. The bone marrow is where our blood and immune cells are made. For a long time, scientists have been trying to create better models to study diseases that affect the bone marrow. Existing models often miss key components, like the diverse stromal cells that support blood cell production. This study introduces a new, more complete model. Researchers developed a novel organoid system called Combo, which combines bone and lympho-myeloid components. This system is derived from human stem cells and cultured in a special scaffold under controlled oxygen levels. They demonstrated that these organoids can be scaled up and can sustain blood cell production over time. They even created chimeroids by adding healthy or cancerous cells to model specific diseases. The results were very promising. The Combo organoid successfully generated all the key cell types found in the bone marrow, including bone, blood, and immune cells. They showed this system could maintain blood cell production over extended periods. When modeling multiple myeloma, they found that blocking MIF signaling reduced inflammation and slowed the growth of cancer cells, suggesting a potential new treatment. This study presents a significant advancement in how we model the bone marrow. The combo organoid system provides a powerful new tool for understanding complex blood disorders and for testing potential new therapies. This could dramatically speed up the development of treatments for patients with conditions like leukemia and multiple myeloma. The next steps will involve applying this model to a wider range of diseases. Up next, our second paper out of 5. A spatially resolved human glioblastoma atlas reveals distinct cellular and molecular patterns of anatomical niches by Pranali Patki and Namita Shah from Amaranth Medical Analytics, Bangalore, India. Let's break down its key contributions. Glioblastoma is a devastating brain cancer, and its aggressive nature is partly due to the complex mix of cells within the tumor. While we know different cell types exist, how they are arranged in space inside a human tumor is still a big question. This study aims to shed light on that spatial puzzle, hoping to uncover new ways to fight this disease. To tackle this, the researchers created a detailed, spatially resolved atlas of human glioblastoma. They combined gene expression profiling across tissue sections with single-cell and protein measurements. This allowed them to map out different cell types and their precise locations within the tumor microenvironment. The team discovered previously less characterized endothelial, perivascular, and fibroblast-like cell states. Importantly, they found a distinct oligodendrocyte population specifically located in the tumor core around blood vessels. This population showed gene expression patterns that were linked to tumor recurrence and poorer clinical outcomes. This detailed atlas offers a powerful new tool for studying glioblastoma. It pinpoints specific cellular interactions within different tumor regions that could be targeted for new therapies. The discovery of the oligodendrocyte population's association with poor outcomes also opens up avenues for improved prognostication and treatment strategies. Up next, our third paper out of 5. Engineered UN1 Cas12F1 for multiplex genome editing with enhanced activity and targeting scope by Yanan Hua and Liren Wang for Frontier Science Center of Genome Editing and Cell Therapy, Shanghai Key Laboratory of Regulatory Biology, School of Life Sciences, East China Normal University, Shanghai, China. Let's break down its key contributions. The CRISPR-Cas12F system holds great promise for gene therapy due to its small size, making it suitable for AAV delivery. However, its current limitations in recognizing specific DNA sequences and its editing efficiency have hindered its widespread application. This research aimed to overcome these barriers by engineering an improved version of Cas12F. To enhance the Cas12F system, the researchers employed a strategy of directed evolution. They screened bacterial libraries and validated promising variants in mammalian cells, ultimately identifying a highly optimized version named EvoCas12F. This engineered system was then tested for its ability to perform genome editing, including in a mouse model, to assess its real-world applicability. The engineered EvoCas12F system demonstrated remarkable improvements. It expanded its PAM recognition to NTNR/NYTR, reducing the distance between targetable sites to just 2 nucleotides in the human genome. Editing efficiency reached an impressive 91%, a 1.4-fold increase compared to the wild type. This enhanced system also proved effective for generating homozygous mutations in mice, even at non-canonical PAM sites. This engineered EvoCas12F system represents a significant leap forward in CRISPR technology. Its expanded targeting capabilities and enhanced editing efficiency make it a more versatile and powerful tool for genome engineering. This advancement holds great promise for developing new gene therapies and for high-resolution research applications. Up next, our fourth paper out of 5, comprehensive cellular analysis with single-nucleus RNA-seq of archived pH gene whole blood samples by Ojasvi Chaudhary and Asaf Rodham from Translational Medicine, Oncology R&D, AstraZeneca, Waltham, Massachusetts, 02451, USA. Let's break down its key contributions. pH gene blood RNA tubes are a common way to preserve blood for gene expression studies. However, until now, analyzing these samples at the single-cell level has been difficult. Most studies have relied on bulk RNA profiling. This research introduces a new method to unlock the potential of these archived samples for single-cell analysis. To tackle this, the researchers compared 2 methods for isolating nuclei from pH gene-preserved blood: mechanical separation and cell lysis. They then performed single-nucleus RNA sequencing. A key step involved using CRISPR technology to reduce globin gene noise. This allowed for a more accurate and sensitive analysis of the captured nuclear transcripts. The results showed that the cell lysis method provided significantly higher nuclei yields, up to 2 orders of magnitude more than mechanical separation. This method also resulted in less biased proportions of immune cells. Importantly, using CRISPR to deplete globin genes substantially reduced background noise, leading to more sensitive gene detection. The approach successfully captured all major immune cell types, including granulocytes, maintaining similar proportions to fresh samples. This study presents a breakthrough method for analyzing archived PH gene whole blood samples at single-cell resolution. It effectively overcomes previous limitations, allowing researchers to study vast collections of samples. Notably, it preserves granulocytes, offering a more complete picture of blood cell populations. This opens exciting possibilities for retrospective research into various diseases and immune responses. Up next, our 5th paper out of 5, RIGI, Adjuvant Immunogen Elicited T-Cell Immunity Controls Trypanosoma cruzi, Chagastic Cardiomyopathy, and Left Ventricular Dysfunction by Nanda Deva Lokagamage and Nisha Jain Garg from A Department of Microbiology and Immunology, University of Texas Medical Branch, Galveston, Texas, USA. Let's break down its key contributions. Chagas disease is a serious parasitic infection that can lead to severe heart problems like cardiomyopathy and heart failure. Unfortunately, current treatments aren't ideal for adults, leaving a critical gap in care. This research explores a new immunotherapy approach to tackle the cardiac risks associated with Chagas disease. To test their new therapy, the researchers used a mouse model of Chagas disease. They developed 2 bicistronic immunogens, BCV and BCVR, which contain key parasite antigens. These immunogens were administered to mice, which were then challenged with the parasite. The study meticulously assessed parasite control, cardiac inflammation, fibrosis, and importantly, left ventricular function. The results were very promising. Both BCV and BCVR treatments significantly controlled the parasite load in the tissues, reduced inflammation, and prevented cardiac fibrosis. Crucially, left ventricular function, which is severely impaired in Chagas disease, was preserved, especially with the BCVR treatment that included the RIGI adjuvant. Importantly, these immunotherapies were found to be safe and non-toxic. This study highlights a significant advancement in the potential treatment of Chagas disease. The developed immunotherapies, particularly BCVR, offer a novel, safe, and effective strategy to combat the devastating cardiac complications. These findings pave the way for future clinical trials aimed at protecting individuals from the cardiogenic risks of Chagas disease.